Hello, my name is Maria Kristanti Alhindi, and today I will present Egg Osmosis Experiment. It's about growing and shrinking egg. I will talk about a little bit background about this experiment. So, what is diffusion? Diffusion is the natural movement of a substance from an area of higher concentration to an area of lower concentration. And how about osmosis? Osmosis is the diffusion of water through a membrane. Some membranes are selectively permeable, meaning that they will only allow some molecules to pass through, usually small molecules. Because H2O is a small molecule, water can pass through a membrane and get trapped on one side because the other particles are unable to get across to equalize the concentration. Both diffusion and osmosis are known as passive transport through the cell membrane because neither process requires energy from the cell. Osmosis is the scientific term that describes how water flows to different places depending on certain conditions. In this case, water moves around to different areas based on a concentration gradient. In biological systems, the different solutions are usually separated by a semi-permeable membrane, like cell membranes. This acts sort of like a net that keeps solutes trapped, but they still allow water to pass through freely. In this way, cells can keep all of their guts contained but still exchange water. Now, talking about our egg, there is a lot of water inside of the egg, but a lot of another things too, like protein and fat. When you place the egg in three different solutions, how do you think the concentration of solute differed between the inside of the egg and outside of the egg? The egg membrane acts as a semi-permeable membrane and keeps all of the dissolved solutes separated, but allows the water to pass through. Here are pictures of osmosis in plant cells and animal cells. So my hypothesis, if I put egg in corn syrup, causes the egg to lose water because corn syrup is hypertonic solution. Egg will shrivel up. And if I put egg in purified drinking water, causes the egg to absorb water because purified water is a hypotonic solution. Egg will grow in size. And lastly, if I put egg in isotonic solution, which I make from 1½ tablespoon corn syrup plus 1 cup purified drinking water, it causes the egg will remain the same size or almost the same size. My strategy after I dissolve the eggs, I make eggs naked. I will put all these eggs in these three different solutions, hypotonic, isotonic, and hypertonic solution. As I'm curious if any difference between organic and non-organic egg, so I run experiment in organic and non-organic eggs too. So for this experiment, I have organic and non-organic eggs, cups and glasses containers which large enough to fit the egg plus liquid, white vinegar, purified drinking water, light corn syrup, spoon, if possible slotted spoon, measuring cups, measuring spoons, 1 tablespoon and ½ tablespoon, sticky notes and marker, and scale. To make the eggs naked, we use vinegar. Vinegar is an acid. Egg cells are made of calcium carbonate. If you soak an egg in vinegar, the egg cell will absorb the acid and break down or dissolve. The calcium carbonate will become carbon dioxide gas, which will go into the air. What left is the soft tissue that lines the inside of the egg cells. It will bounce. The vinegar dissolves the cell of the eggs and they become translucent and water is able to pass through them. The eggs have become an easily observable example of cells that are semi-permeable. So what I did on this experiment, I placed egg in each cup, pour in enough vinegar to cover each egg, and the bubbles will start to form around the egg, and it will float up. To keep eggs submerged, you may put butter knife in the cup to hold it down or anything that can make the egg stay down and allows it for 24 hours. The next day, gently holding the egg in the cup, pour out the old vinegar and replace with fresh vinegar and let sit for another 24 hours. You may repeat this process until the cells are fully dissolved and only the membrane remains. In my case, it took me 3 days to dissolve this. Gently remove the eggs using the salted spoon and rinse with tap water in the sink. Then gently put the dissolved egg aside for a moment on a plate. So for this experiment, as I watch for organic and non-organic eggs, so I prepared 3 different sugar water solutions for each organic and non-organic eggs as follows. I label first container hypertonic and I pour 1 cup corn syrup. Second container I labeled isotonic by adding 1½ tablespoon corn syrup to the 1 cup measuring cup and fill the remainder with the purified water and stir to dissolve. And the third container I labeled it hypotonic. I pour 1 cup of purified water. I do the same thing for organic and non-organic eggs. And I weigh the dissolved eggs before I put to each container. Gently put one of the dissolved eggs in each of container. Wait for 24 hours. So as mentioned before, I'm curious if any difference between organic and non-organic eggs. So I ran experiment into these 2 kinds of eggs. After 24 hours, gently take the dissolved eggs out of the container and weigh it again one by one. So here are the results of the experiment organic and non-organic eggs. It's interesting how organic and non-organic eggs in these 3 different kind of solution, they both have the same percentage change in weight. Like in hypertonic solution, both organic and non-organic eggs, it has a negative 29% change in weight. And for isotonic, there is no change of the weight as that what we expect is 0% there for both eggs, organic and non-organic eggs. And for hypotonic solution, even the start weights of the eggs for the organic and non-organic eggs different, but at the end both have the same percentage change in weight, which 9%. Interesting, right? So it's no matter organic or non-organic eggs, when we put on these 3 kind different eggs, different solution, they both have the same result. I still don't get it on this. I really want to find out why or how come there is no different percentage change in weight. Or maybe I should have a different kind of scale with showing more accurate in weight. But I don't have that kind of scale, so I just use the scale that I have. And this is the result from it. So as I mentioned earlier, it's interesting there is no different result between organic and non-organic eggs. Even when the start weight of the eggs different, at the end, the percentage of change in weight is the same result. So in this case, when we have the hypertonic solution, eggs shrivel up. And in the case of the isotonic solution, eggs stay at the same size. And lastly, in the case of the hypotonic solution, eggs grew in size. Really, I still don't get it why there is no difference between organic and non-organic eggs. So as conclusion, water is hypotonic to the egg. The egg will gain mass over 24 hours. Corn syrup is hypertonic to the eggs. The egg will lose mass over 24 hours. So before I ending this presentation, I'd like to give little information about osmosis and us. Every cell in our body needs the right amount of water inside of it to keep it safe, produce energy, get rid of waste, and other functions that keep us healthy. This is why medicine that are injected into patients need to be carefully designed so that the solution has the same concentration of solutes as their cells, which is isotonic. If you were sick and become dehydrated, for example, you would get a 0.90% saline IV drip. If it were too far off from this mark, it wouldn't be isotonic anymore, and our blood cells might shrivel up or even explode, depending on the concentration of dissolved solutes in the water. Osmosis works just the same way in our cells as it does in our egg cell model. Thankfully, the semi-permeable membrane of the egg is much stronger, so we don't have to worry about the eggs exploding. We don't want to make a mess. However, still, we have to be careful as we don't want to make a mess during our experiment. Thank you for watching and listening to my presentation. I hope you enjoyed.